Today we'll learn about the central dogma of biology and Francis Crick's adapter hypothesis. Let's start by understanding what the central dogma is. It's the process of using the instructions in the DNA and converting them into a functional product, like an enzyme or another protein. Looking at this diagram from Genetics: A Conceptual Approach, we can see all the steps included in this mechanism. Today, we're mostly interested on the last step, translation. This is where the fun actually happens and where the genetic information turns into functionality for the body. Here's some key points. What are we reading? The information in this step is read from the nucleic acid sequence of mRNA or messenger RNA. mRNA is created by mirroring out a specific gene in the chromosome, so its base sequence complements those of the DNA strand. Each trio of bases in the mRNA is known as a codon. How do we read it? We read this information using rRNAs or ribosomal RNAs. rRNAs have two lobes, a small one and a large one, and their main function is to facilitate the production of proteins based on an mRNA. How do we know what amino acid is related to each codon? This is done through tRNAs or transfer RNAs. Each kind of amino acids has their own tRNA to bind to. Each tRNA has a three-base sequence known as anticodon, and they are complementary to the mRNA sequence that calls for that specific amino acid. Why amino acids? tRNAs mainly connect to amino acids because they are the monomers that make up proteins, hence why they are important in this process. What about the adapter hypothesis? Francis Crick used this hypothesis to try and explain how one can go from a sequence of nucleic acids to an amino acid chain. Let's use outlets to think about this. I want to be able to use my Brazilian straining iron in America, but it's easy to see that the plug won't fit what is on the wall. So to make it work, I can use an adapter, the tRNA, that will match the holes on the wall, or the mRNA sequence, and will fit the pins of my hair straightener, or the amino acid. Using the adapter, I can plug my hair straightener in, just like we use the tRNAs to specify which amino acid is encoded by each codon. However, if a mutation occurs on the DNA responsible for coding the tRNAs and consequently changes the shape of the transfer RNA, that would lead to problems in the production of proteins. The tRNA matches perfectly the inside of the ribosomes. It also lines up the anticodon correctly and has a region responsible for connecting the amino acids. If the shape changes, then we wouldn't be able to use tRNAs as adapters, and the cell would no longer produce proteins. This can also be seen using the plug analogy. If the shape of the tRNA or adapter changes, then we can no longer connect the hair straightener to the wall outlet, and I won't be able to use it.